So this talk is going to be slightly different from you have seen so far in this summit. We're going to shift the gear a little bit from towards science instead of a spark. We're going to show you some of the works that health and life sciences team in Databricks in collaboration with Harvard University and Broad Institute. And I hope at the end of this talk we can generate enough interest and excitement that will encourage more of you machine learning engineers to join the fight and win the battle against devastating diseases. So with that, I'll start with this quote from Freeman Dyson, who is a great physicist and a mathematician. He once said, "The deepest concepts in mathematics are those which link one word of ideas with another." So this is especially true when we think about biology, mathematics, and statistics. For example, the genetic algorithm, one of the optimization techniques, is closely related to evolution theory. And L1 lasso, in statistics, L1 lasso and grouped lasso are either motivated or closely related to the genomics analysis. Now, none of them is perhaps as deeply connected as artificial neural networks and human brain. So this figure is an illustration of neurons. We have roughly about 120 billion of such neurons in our human central nervous system. And all these neurons are connected via this protruding, do I have this? So I'm going to use the mouse. So via this protruding nerve fiber, through this nerve, what do they call axon? Through this axons, the neurons are constantly communicated with each other. And whenever there's a stimulus like a sound, a visual, a sensation, a neuron will activate and send over electrochemical signals along the fiber. These signals are going to be gated when they enter the cells via either biophysical or chemical processes. But in the end, they're going to be relayed to the next neuron or next sets of neurons. And the entire process can actually be abstracted out with a very simple mathematical formula. So you have a vector of random vectors essentially coming from different wires, and they're weighted differently with different weights. And they're summed up inside of the cell body with some adjustment after some transformation function phi, and if the totality of the signal is above a certain threshold, the neuron will activate and sends off the signal to the next neuron. So this is how you actually see in the deep neural networks, you get all these functions called activation function. Now, we said there are over 120 billion of such neurons in our brain, central nervous system, and each neuron is actually connected with as many as 10,000 neurons. And this figure shows you at the system level how our human brains actually orchestrates and processes all these signal cascades in 3 different functional states. On the left, at the resting state, you can see the connectivity is the connections are sparse and the energy is low. But in the middle, at the attention state, you observe the increased connectivity and higher energy. But at the memory state, you do see this much, much higher connectivity and the energy state. That's suggesting a very high capacity learning process is happening. Now, a little bit background about this artificial neural networks. It actually really started as early as the 1940s, but in the 1960s, especially in the 1980s, it generated a lot of excitement. Unfortunately, it was not as successful at that moment for many reasons. Not enough data, not enough computing power, but perhaps most importantly, it was too advanced of its time. Now, it wasn't until the mid of the 2000s, Jeff Hinton from University of Toronto and others did some pioneering work, really resurrected the artificial neural networks from what is called AI winter. But many people didn't realize it was the first time some students from Jeff Hinton's group first applied deep neural networks to model the chemical properties of a drug to its efficacy and safety profile. And their team actually won the top prize in this Kaggle challenge study hosted by Merck. And the point here being the deep learning was actually first taken back to its very root and demonstrated its power in solving some challenging life science problems. Now, fast forward 5 years to this moment, deep learning has been successfully applied in several areas, especially in image analysis, in diagnosing the several diseases such as melanoma, diabetic retinopathy, and abnormalities in heart. Now, especially the third example on the right, this group from artery, which is essentially from Harvard, also from Stanford. The important part of this is they actually won the FDA approval to be used in real clinical setting. This is a very, very significant step forward for deep neural network because it essentially pushed the boundary of medicine. It essentially making a software as a medical device. This is completely revolutionary. Now, we have seen most of these examples are actually related to image analysis. We have seen, but we know that deep neural networks has a lot of potentials, can do a lot more than what we do today. So we have seen how deep neural networks can be used to model the chemical properties and the structural, chemical structures and the activities. We have seen how deep neural networks can be applied to improve medical image analysis results. And now a big question is, can we actually leverage deep neural network to identify those fundamental genetic factors that are disease-causal, and so that we can actually treat those disease at its root level per individual patient. So with that, I'd like to hand over to my collaborators, Yishang, who is a leading genetic scientist from Harvard University and Broad Institute. He's going to walk us through some of the challenges and the pioneering work that his team is engaging on in this space.
Good morning. I guess the microphone is working. Thank you for the introductions and invitation for me to talk about my research here. My name is Yishang Xu, and I am the director at the Genomics Center at Harvard Medical School, and I'm also the associate professor at Harvard Medical School and Broad Institute. As you know that I'm a biologist, so in the following 20 minutes, I'm going to talk about the hardcore biology question that we are facing, and sit back and relax. And I'm going to direct all of the engineering questions to Yang. All right, so in the past 10 years, there is a great progress in the human genetics area, and especially the Human Genome Project completion in 2005. Everybody know what Human Genome Project is. Basically, it's a project in the biomedical field that is comparable to the Apollo project, which spent 10 years, multinational project, spent $1 billion to sequence one individual's genomes. And since finished that project, then we know the position of each genomes that allow us to put the sequence markers into a chip and allow us to measure the genomics informations. And as up to date that people using the Genome-Wide association study approach, and scientists has already identified roughly about 13,000 of the genetic variations that associate with roughly about 2,000 of human diseases. And shown here is the chromosomal positions of those associated genetic variants as shown here. And the different color indicate the association with different diseases. If you are interested, you can go to the NHGRI GWAS catalog website or the EBI, which is a European bioinformatics database, to check it out. As shown here, that you can see that this associated genome actually can be found in almost all of the autosomal chromosome as well as the X chromosome. However, you can see that it seems there is nothing to do with Y chromosome, right? And you know that Y chromosome is in men only. And based on the evolution study, that we know that Y chromosome actually is disappearing. And based on the evolution study result showing that there are 300 genes actually disappear in the Y chromosome. So I guess one day men will become extinct species. So basically the implication of this is a majority of human protein coding gene. And now we know there are about 20,000 of human protein genes. And majority of them probably will involve in pathophysiology of human diseases. And the human protein gene, the number of human protein gene is still evolving because we still have a lot of unknown in the human genomes. And talking about the concept of the genome-wide association study, actually it's a very simple and straightforward. As shown here, the idea of the genome-wide association study is to systematically compare the difference of the DNA sequence one by one, and try to identify the enrichment of the DNA sequence in certain positions that are enriched in the diseased patients compared to the healthy control. And these potential genetic variations, it could be mutations or polymorphisms that may change gene functions and they increase the risk of diseases. So a typical genome-wide association study design involves in collect about 10,000 to 100,000 of the subjects, including both of the diseased patients and the healthy control. And then we collect their blood DNA, and then we measure their genetic variations by either using the gene chip that may have about 5 million genetic variation into the gene chip, or recently using the next generation sequence technology, such as whole genome sequence, to ascertain 3 billion of the DNA coding in the human genome. And again, then we compare the difference between the diseased patients and in the healthy control and try to identify the enriched of DNA coding, and then using this to identify the genetic variations and genes that may affect diseases. So why this is kind of a study is important. Based on several recent observations, people have reported that the percentage of the successful FDA approval drug are actually correlated with the availability of information that are available for human genetics. Just give you an example here, a study published 2 years ago in the Nature Genetics. They find that the FDA approval drug with human genetic information, such as the OMID genes, which is the mutation in the coding regions that cause diseases, as well as the GWAS gene, which is the polymorphism in the non-coding regions that may associate with diseases. They find that with the human genetic information, the FDA approval drug are 5 to 10 times more likely to be successful compared to those drug targets that are not approved by FDA. On the other hand, the failure target at each drug development stage, such as preclinical, phase 1 and phase 2 and 3, are more likely to be those targets without genetic variations. That means that the impact on medical care from this type of human genetic study could potentially be substantial. As many of you know that the R&D spending on new drug development actually for past 20 years actually doubles from about 20 billions per year to 50 billions per year. However, the approval drug per year actually did not increase, as shown here. And on the right-hand side, this figure is showing that the successful of the drug targets at a different stage across the year, they actually stay the same or even decrease. So what does that mean? That means the pharmaceutical company need to have a better drug development pipeline. And based on the previous evidence I just present, it looked like that utilizing human genetic information and using human genetic information to validate drug target could be the key to increase the success of the new drug development, as well as to save the cost, average cost for the drug development. With that in mind, I just mentioned that there are more than 30,000 of the genetic variation has been found to be associated with diseases. You might imagine that the pharmaceutical companies probably are very busy to pumping their pipeline to get their drug approved. However, unfortunately, this is not the case. And the reason is 95% of these so-called diseases-associated genetic variants, they are located in the non-coding protein regions. And if you still remember your high school biology, the non-coding region, including the intron, as you can see here, the color in green, intron of the gene non-coding into the protein, also including the DNA regions that in between two genes called the intergenic regions. And these regions is so-called the non-protein coding regions. They used to code the junk DNA with unknown function. That's very difficult to translate finding into clinical use. Just to give you a couple of examples. Shown here is a regional plot of association between genetic variant and the motion sickness. And on the right-hand side, y-axis is the negative log 10 association p-value between the genetic variant and the motion sickness. And the higher the negative log 10 p-value, the stronger the association is. And the x-axis is the genetic coordinations, which is the one-dimension physical locations of each of these genetic variations and the genes on linear DNA sequence. As you can see that there are strong genetic variants associated with motion sickness with p-value lower than 10 to minus 15. It's called the 10 to minus very exciting. However, you can see that all of these genetic variants, they are located in the gene desert regions. There is no protein coding gene map into these regions. Therefore, we don't know which gene to target to treat the motion sickness for this case. On the other hand, another case is there are too many genes in the regions. Shown here is the genetic variation associated with the risk of osteoporosis, which is a disease in the elderly, and they will increase the fractures risk and also increase the mortality. As you can see that very strong association between genetic variants and the risk of osteoporosis is shown here. However, if you map to the protein coding gene, you can see that there are so many protein coding genes here. Therefore, we don't know which one or which them are the underlying causal genes. Therefore, we don't know how to target this osteoporosis. Another example shown here, a group of tightly clustered genetic variants in the intron of FTO genes. They are strongly associated with risk of obesity. And this so-called FTO locus, which is the most widely studied genomic locus for the obesity. And as showing here, you can see that very tightly clustered into the FTO genes. Then can we just say that FTO gene is the underlying causal gene? And that means this genetic variant will sort of change or affect the FTO gene functions and then increase the risk of obesity. Is that the case? Maybe not necessary. I'm going to show you an example that we did to prove that FTO is not a gene. In order to identify what's the underlying causal gene for this genetic association with the obesity, we actually conduct a functional genomics research and to identify the underlying causal gene. Shown here that using the functional genomic research, we find that this obesity-associated genetic variant, they are located on the active enhancers predicted by the histone proteins. You probably cannot see it clearly, but trust me, basically this active enhancer is highlighted in yellow color, and they only show on fat-relevant cell and tissue, such as the adipose progenitor cell, as well as a mesenchymal stem cell, suggesting that this region probably involved in gene regulations in the tissue-specific manners, and that tissue-specific manner actually relevant to the diseases itself. In order to understand what's the downstream genes that this active enhancer actually affects, we conduct a three-dimensional genomics using three-dimensional genomic technology such as chromatic conformation captures, and we construct the three-dimensional genomic interactions between this FTO-associated genetic variant with any genomic regions on the same chromosomes. And using this so-called topology association domain content matrix plot, I'm going to talk about it a little bit detail later, we identify that there is a gene called the IRX5, which is physically interacting with these associating regions. And the IRX5 gene actually is quite far away from this FTO sort of associating variant. It's about 2 million base pair away from these regions. In terms of the DNA sequence distance, this is very far away. So we hypothesize that this is probably the so-called long-term gene regulatory sort of mechanisms. And basically, this finding also confirmed by expression QTL analysis. I'm not going to talk about the detail. Basically, the study has been published last year in New England Journal of Medicine. So basically, our hypothesis is here. The obesity-associated genetic variant located in the FTO gene, actually they are located at the active enhancer regions as shown here, and there are multiple transcription factors binding on it. So in normal conditions, the transcription factor will binding on it and then enhance the IRX3 gene expressions. However, in the obesity subject that carry such genetic variations, because their mutation and polymorphism change the DNA sequence in these regions, that the transcription factor cannot bind on it, that inhibit IRX5 gene expressions and then increase the risk of obesity. In order to test such a hypothesis, we actually conduct the gene editing by CRISPR-Cas9, which is the gene engineering way to directly editing your DNA sequence in your living cell. And then we do the gene editing in the human adipose from the obesity subject that carry this risk allele. And just going to briefly present the result. As shown here, the y-axis is the gene expression in the fat cells in those obesity subjects. When we replace their risk allele in these regions to the wild-type allele, we observe that the IRX5 gene expression increase. And when we replace this risk allele back to the wild-type allele as they originally have, and we find that their IRX5 gene expression back to the normal, which suggests that this region actually regulates IRX5 genes. We do the same for the FTO gene. We did not find any gene expression change. So that suggests that FTO gene is not the underlying regulated genes. In the meantime, there are other studies also doing similar research. They found that this obesity-associated variant physically interacting with promoter of IRX3 genes as shown in this circle plot. This is IRX3 gene and this is FTO, but not the FTO and IRX5, which we find IRX5 in adipose, but they find IRX3 in mouse brains. So basically, based on this result, suggests that the gene regulation actually is tissue specific and it's based on the regulation mechanism will be different in different cells in your bodies. And also the gene regulation element is interacting with your gene promoters based on their three-dimensional space in the physical proximity and by the DNA looping mechanism. And shown here is a cartoon for the DNA sequence. Assume there are two enhancers, green enhancer and blue enhancer here. In terms of DNA one-dimension sequence, you can see that this green enhancer is, in terms of their distance, is more close to the gene compared to the blue enhancers. However, in the living cell of the brain, we observe that actually this type of the blue enhancers physically interacting with the promoters of the gene and physically more close to this promoter of the genes in terms of the three-dimensional space compared to the green enhancers. However, in the living cell of the different tissues, such as the limb, and we find that the green enhancer actually is interacting with the promoter of the gene and is physically more, in terms of three-dimensional space, is more close to the promoter of the genes to regulate the gene expressions. So based on this evidence, it suggests that when we're looking at the gene regulation, we shouldn't looking at the linear distance of one dimension of a DNA sequence. We should looking at the 3 dimension of the DNA structures in the living cells. I just mentioned that the 91% of the diseases-associated genetic variants, they are actually located in non-protein coding regions. And I just give you an example to prove the concept that we can using the functional genomic research to identify the underlying causal gene for those non-coding associated variants. And the question is, can we simultaneously to test all of the 13,000 of genetic variants at the same time and to identify their underlying causal variant, not just doing one gene at a time? And the answer is yes. With the advance of the technology and with the advance in the analytical approach, such as using machine learning methods, that will allow us to perform such analysis. And then, oh, my time is up, so I'm going to speed up. And then to identify the underlying causal gene for such associated variant. And one of the methods that we use is so-called Hi-C sequence. Hi-C sequence is a genome-wide version of the chromosome conformation captures and to identify DNA loopings. And what the Hi-C sequence data do is basically we're using a chemical to cross-link the interacting DNA regions. And then we're using the restriction enzyme to cut this interaction region out. And then we sequence them. And then we're using these sequence interactions to construct so-called the contact matrix. And shown here is a heat map of the contact matrix. We call it as a contact map. And using the contact map information, as you can see that the contact map actually displays the density of the interaction among pair of the genomic regions. And then using this contact map information allow us to identify root informations. And using this root information allow us to identify the enhancer promoter pair and then identify the underlying regulatory genes. However, the Hi-C prediction is not going to be perfect because it's a sequence technology. So basically, it could be due to the accuracy could be due to the sequence error and could be due to the mismatch of the restriction enzyme cuttings. Also could be due to other interactions such as enhancer, promoter, and physical interaction. So how do we increase the accuracy of the predictions? So basically, what we can do is we need to have a lot of genomic informations to improve the predictions. Also, we are using the machine learning technologies to integrate this information together. For example, to build the tissue-specific gene regulation circuits, not only we need Hi-C, we also need a taxi sequence, which is to detect the open chromatic regions, ChIP-seq sequence, which is to detect the function of open chromatic regions, such as enhancer, promoter, suppressors, and the repressors, as well as RNA sequence to identify the real-time transcription information, as well as transcription factor binding site to identify the active enhancers and the upstream regulators. So what we do is we are working on a project to perform experiment as listed here. And basically, this involves in some sequence experiment. I'm not going to talk about the detail. However, I would like to highlight, in order to get a very high resolution data, we actually have to sequence up at least 2 million of the sequence read. That is equal to 2 terabytes of the data per sample for this Hi-C experiment in order to get the resolutions of the information that we need. And we perform this experiment on the normal human primary cardiofibroblast myocytes from the atrioventricular and as well as the primary skeletal muscle, along with publicly available data showing here. And then we actually put all of this data, there are about hundreds of terabyte data, into a deep learning machine learning models implement in the TensorFlow notebook to predict the enhancer promoter gene pair and then to predict the underlying causal genes. And the training set are obeyed from the VISTA project, which is the including about 1,600 enhancer promoter gene pair that function or validate to have a regulatory relationship in mouse model, along with another 1,200 enhancer promoter pair without regulation relationship. So the analysis is still ongoing, but hopefully with the high throughput parallel computing resource that under the cloud, we will be able to finish the analysis in a short time. And in the future, so we will build a browser and the database and make it publicly available for people to search the findings. In summary, I have talked about and then provide a proof of concept that using functional genomic research will allow us to identify underlying causal genes from the non-coding association variants from the whole genome sequence or genome-wide association study. I also show you that our ongoing study using machine learning as well as large-scale genomic structures data to improve the predictions of the enhancer promoter structures and then to predict the underlying the genes. And hopefully these underlying genes will be able to use as a drug targets to treat the cardiovascular diseases. Thank you for your attention.
Thank you very much, Yixing and Young. I think we're at time, so if you do have any questions, you guys will be around for a little bit.
Sure.
Feel free to come up to the front or in the hallway, and you can ask your questions. Otherwise, we're going to get it set up for the next session. Thank you very much.